Good morning, everyone. You are welcome to my YouTube channel. The topic for discussion is the alternative pathway of the complement system. The complement system is an integral part of the non-specific immunity or innate immunity. The complement system is made up of 30 serum proteins that are regarded as proenzymes. They are inactive until they get a signal. For the alternative pathway of activating the complement system, it starts by spontaneous cleaving of the C3 component. The C3 component is cleaved into two fragments, C3A and C3B, by a blood enzyme. Now, this becomes stable only when it binds to a lipopolysaccharide, LPS, of the gram-negative bacterial cell wall or any other type of endotoxin. So the factor B is a blood protein which adsorbs to bound C3B. What is C3B bound to? The LPS, lipopolysaccharide of the gram-negative cell wall, and is cleaved into two fragments by factor D. This leads to the formation of the active enzyme C3 convertase, which is C3BBB, and is further stabilized by a second blood protein, properdin, which is changed or converted to C5 convertase, C3BBB3B. This convertase then cleaves C5 component to C5A and C5B. C5B is the larger fragment which will bind to the complex and then follow rapidly by C6 and C7 components. C5B, C5B, C6, C7 complex has an unstable membrane binding site, but becomes stable once binds to a membrane. Then C8 and C9 then bind, forming the membrane attack complex, which creates a hole in the cell membrane of the target cell. Now, lysozyme is known to move in through the peptidoglycan cell wall after passing through the outer membrane of the gram-negative cell wall. Here is the flow chart showing the alternative pathway. Quickly, the C3 component is cleaved into C3A and C3B fragments. The C3B is unstable until it binds to the lipopolysaccharide and it becomes stable. Following that, factor B adsorbs to the bound C3B and is cleaved into two fragments by factor D. This leads to the formation of the C3 convertase. Properdin binds to the C3 convertase, that is C3BBB, and is changed to C5 convertase. The C5 component is cleaved into C5A and C5B. The C5B being the larger fragment binds to C6 and C7, although it's unstable initially until it binds to a membrane binding site. Once it binds to a membrane, then C8 and C9 bind to the complex and a pore is formed. What are the functions of the complement components in alternative pathway? One, inflammation. You have two, elimination of immune complexes, phagocytosis, neutrophil activation, through a process known as chemotaxis. It's like calling for more help to eliminate the foreign material, which is an antigen. Thank you for listening.